Should I use it? It's probably recording.
Yeah.
All right. Is this on?
You don't need slides.
Oh, yes, I do.
Jordan, can you help me with the mirroring on this thing? Thank you.
Shot of Andromeda.
Well, I can get started. Okay. So today I'm going to talk to you about, I do a lot of indoor bacterial work, microbiology. I started with Norm Pace years ago doing shower curtain microbiology, which I still get a lot of grief over. I'm glad to be at a conference where that's no longer the case. And I wanted to tell you that a shower curtain is a perfectly good ecosystem. All right? The NSF reviewers aside, mocked me on it for another proposal entirely. So I'm really glad to see some respect. No more Rodney Dangerfield. All right, let's see what we got. Great. All right, thank you very much. So I'm going to talk to you work that I have tried to do on viruses in the indoor environment. Actually, some really interesting results, but well, you'll just see. All right, so environmental viral diversity sensu lato is amazing. Biodiversity is largely unknown planet-wide, or we're starting to get to know it. But it's really quite interesting, and there's a lot to know. There's an estimated 10 to the 30 bacterial cells on the planet, and there's about 10 times that, the number of viruses. That's 10 to the 31. Some perspective on 10 to the 31, I believe it's the diameter of the universe in millimeters, all right? So we're talking a very, very big number of organisms, all right? Indoor microbiology, indoor diversity is almost completely unknown as well. And I find that pretty interesting, and I'm really encouraged in some ways, or discouraged, to see that no one has actually presented yet on viruses in the indoor environment. So, at this meeting, right? So I think I really am trying to break new ground. Marine systems have been really well studied. We have sort of the leader in the field in my department, which is the reason I thought I could even do this in the first place, because I had someone to talk to, right? And to help me along, because he really is the world expert. Marine systems, animal systems, oral, gut, lungs, these are places loaded with viruses, loaded with unknown viruses. And so it's really interesting, of course, to see what is happening in an indoor system, because there's going to be viruses there too, you just know it, they're everywhere, right? And if we're bringing bacteria into this environment, we're going to bring all of their viruses, and we're going to have viruses of ourselves that we're shedding all the time. And how does it get from person to person? We really don't know. We do know, in some cases, for very well-known eukaryotic viruses, but there's probably a lot of unknown eukaryotic viruses, and then there's a whole bunch of phage that we have no idea what they're doing. Turns out that they're much more interesting for human health than we also thought. So they're definitely killing the bacteria, but they also might be important for our immune system. All right, so that's what I'm going to look at. All right, so I wanted to start with a study that I did earlier with Maya Breitbart and with the Rohr Lab. First, to show you an example of a virome study that actually was quite successful, which shows that I can actually do this kind of work, and which will be important later on in the study. And it's much more actually relevant to this conference than I previously thought, because this is actually, what this is a viral diversity study of the infant gut starting at time 0 in the meconium, and then we after a week later and another week. So we purified all the viruses out of it and looked at them as it changed over time. And actually, it was a massive, massive change from one week to the next. I mean, there was almost no viruses after the first week that were there in the second week. So when people were talking about infants and talking about microbiology of infants, you really have to start thinking about the viruses as well. And I'd love to come talk to you about it. My colleague just put out a study in PNAS that said that there are 4 times the number of viruses in the mucosal lining of the intestine than there are in the gut, stool itself. And it's a hint that they have something to do with prepping the immune system for the bacteria. So if you're going to look at the development of the microbial community in the infant gut in the hospital, it might be nice to know what the environmental viral diversity is when you do that. And it is really possible to do that. So let's talk about what we found in the study, and it'll prep you for what I'm going to be looking for. First of all, I wanted to show you a nice little picture here of some of the viruses in the gut. So when we used microscopy and we filtered out the bacteria, we found all those little dots there, look like stars in the universe. Those are all little specks of VLPs, viral-like particles. They're the right size for viruses. We don't know that they're viruses, but they probably are. And it shows this enormous density of these, and the ratio is typically 10 to 1. 10 viruses to 1 bacteria in most environments. Marine systems, gut systems, I think soils, loads of systems, 10 to 1. Keep that number in mind. I'm going to talk about that in the indoor session. I also want to show you that there's beautiful unknown phage and viruses in there that we don't know anything about. We got this weird tail-like virus here. These are some beautiful TEM pictures that Mike Breitbart did, one of the hardest working graduate students I've ever met. And all these real circular viruses might be eukaryotic. And another thing I'm just going to point out, and this is very typical of these settings, is we don't really know anything about them. So in this setting, when we blasted, just looked at proteins that we had that had anything to do with viruses, something like 65 to 70% had no hits to anything. This is really important to know, because if you want to tell what a virus is, you got to be careful, because a random bit of DNA could be anything. So how are you going to make sure that that random bit of DNA is not some piece of a bacterial genome of a bacteria that you don't know? How is it really a virus? All right. So keep that in mind as well. Okay, so environmental viral diversity, just to reiterate, there's lots of viruses in the environment. The viral-like particle ratio to bacteria is typically 10 to 1. It's very high. Molecular methods, the majority are unknown. When we look in the molecular methods, we don't know anything about them. There's loads of them in the gut, water, soils. That's also important, because I decided to look at restrooms to start this project, because we have a very good study by Flores et al. In Rob Knight's lab that had already looked at the bacterial diversity in the restrooms and found that there's some settings which had bacteria from all these various sources in one place. So it's a kind of a nice place to look if you're really interested in looking at diverse viruses in an indoor setting. Okay, so the built environment viral diversity. So the basic questions that I want to get is the question that you just have to start with. What's there? What's the abundance and diversity of viruses in built environment settings? So I'm going to start with the restroom, and I'll talk about a little more why. What are the diversity of phage? I expect to find mostly phage. It may not all that. I hope to find eukaryotic vice as well, but they're going to be the most abundant. Can we find any human eukaryotic vice? These are all the things that I want to address with this study. Can we find these eukaryotic viruses? Because ultimately, there's a very interesting part of the study. We want to know how humans may be passing viruses among each other. Are they shedding them constantly into restrooms from otherwise healthy people? What's the ratio of viruses to bacteria? We know it's 10 to 1 in every other setting. We can get at this question. And are there associations between the bacterial diversity, for instance, and the types of viruses that are in there? We know there's strong associations in other settings. We're going to get Greg Paparazzo on some software to compare viral and bacterial diversity, and usually there's a really tight correlation in many settings between what the viruses are and what the bacteria are. So can we answer these questions? That's what I went into. And I quickly found out that the real question I wanted to ask is, can we get any data at all? Right? Because the biomass is really small. I mean, it's really small. I mean, you go into marine systems and guts, you can get a lot of viruses. You go into this, swab off a table surface or a bottom of the restroom or a soap dispenser, yeah? What are you going to see? There's not hardly any bacteria there. Are you going to get enough DNA to do this work? All right, because it's worse than just getting DNA. That's the easy. All right, so I'll go back, and we talked about this before, and we've gone through it over, but there's a lot of engineers here. What do they know, really? Microbial diversity. Just kidding, my dad was an engineer. I just like to get engineers forever. Studies, of course, culture-dependent techniques are passé. Well, not really passé, but they do miss a lot of environmental viruses. Bacteria, they're going to miss the viruses as well. You can culture viruses, the ones you know, very easily, but we want to find the ones we don't. So the culture-independent molecular methods, the ones that I mostly worked in Norm Pace's lab and in my own lab, are based on the amplification of a single gene that all of the cells have. So all cells have ribosomes, all cells have to make proteins, they're all going to have ribosomal RNA, small singular RNA. We have a huge database of them, we have primers to them so we can target them in lots of environments. That's great, so we can go and get these guys wherever they are. Wonderful. But this is not true in viruses. There's no single gene for all viruses. They're very small. They don't have a ribosome. They use yours. They use the bacteria's. So there's no gene that you can simply target with PCR primers. So what are you going to do? Okay, so you can get some genes that will target groups of viruses, and people have done that very successfully in marine systems. But I don't know what's in the system. I don't know what I'm going to miss. I want to get them all. How am I going to do that? So the unknown viruses, we're going to use a different method, which we have talked about, a little bit of shotgun metagenomics. Instead of going after a single gene, we're going to go after all the genes. All the genes. Because we don't know what's going to happen. We just got to sequence DNA and see if we can stitch it back together and find out what the viruses are. Okay? In order to do this, of course, well, DNA is DNA. Bacteria have DNA. We have DNA. Fungi have DNA. Viruses have DNA. At least DNA viruses. That's what I'm going to target first. RNA viruses, we're just going to forget about them. They're very important, but it's we start with DNA. There are herpes viruses are DNA. Lots of them, human DNA viruses, and of course, phage are DNA viruses for the most part. Okay, so in order to figure out that it's actually viral DNA, you have to purify the viruses. You have to. Otherwise, you just get bits of DNA. Most of it is going to be bacterial. So you get out the bacterial DNA, right, and you get the viral-like particles. We're going to select by size. Then you extract that. Then you know it's a virus. All right? Even if you have a little contaminating bacterial DNA, it could be a problem because there's things called prophages. Prophages are integrated viruses, right? And there's also, so you want to know it's freely, but there's also lots of viral genes that have gone horizontally into bacterial systems. So you have to get rid of the bacteria, which turns out to be an enormous problem. All right. So this is the thing. First of all, you've got a problem. Bacterial DNA is like 100 times bigger, right? They're really big genomes. Viral genomes are small. So you just have more DNA. You've got to get rid of them. What's free living and not a prophage? You've got to know that. And which sequence is actually viral. Right? You have unknown sequences there. So you have to make sure it came out of a viral capsule to say that this unknown thing that matches nothing is actually a virus. Right? So that's really the trick. And it's all been perfected in these other systems. Okay, so just a little background on, this is a slide from Jack Gilbert. If you look up here, we've got the marine, this is a marine system, but I added little viruses here. Jack, do you like that? No, he's not here. Yeah. All right, so you have all the viruses here, which you have to purify away the bacterial cells. You let with the virus cells, you explode the viral capsules, right? You take out the DNA, you break it into small little chunks, and you sequence all the chunks massively, and then you start to stitch those genomes back together using bioinformatics. You blast them, you figure out what they are, and that's essentially what we're going to do. Okay, so the restroom I thought was a good place to start in this project. Several reasons. It's a shared space for clear potential for sharing viruses, disease or otherwise, you're going to share things. Secondly, there's a previous study that was already done. It showed that there's extensive bacterial diversity in restrooms. There's strong surface-associated patterns. You see differences on the floor, the toilet seat, and the soap dispenser. All right? Very clear differences. There's multiple input sources, skin, gut, and soil, especially on the floor, which is where I'm going to focus. And it's a good place to find diverse communities. All right? So if I want to find lots of different viruses and up my chances, let's start there. Okay? I think it's 5. So the sample collection that I started, I actually did a bacterial study along with this because I knew this was sketchy, but I also wanted to know what bacteria were there so I could correlate the viral diversity and the bacterial diversity. So I had 4 restrooms at SDSU, 2 on my floor, 2 on the bottom floor, 2 male, 2 female, 2 high flow, and 2 low flow, like flow. That sounds like use, high use, low use, and we measured that. I sampled, for the bacterial study, I sampled all these 3 environments. The reason I chose these 3, they'd already shown that they were the most different and most diverse. All right? And we sampled these 3, and I'm going to focus on the floor because it had soil, gut, and skin, and everything. All right? So it's going to be the most diverse, and it's really easy to get loads of surface area because it's flat, tile, you just keep going like this. Okay? So you can get lots in your little swab. You like my swab? All right, so how do we do this study? We actually did it in kind of a funny way. I thought I was doing a time series. It turns out I was just doing the same thing every day, but it was pretty cool. What we did is we bleached the crap out of it for 20 minutes. That's how we got rid of everything. I mean, it took 20 minutes to see not a single particle under the microscope. We sampled it 8 hours later, so we let it build for 8 hours and then sampled it. We confirmed bacteria and viruses under the microscope using epifluorescent microscopy, which turned out to be really nice, had our best results actually. We extracted DNA for PCR, did all that. Illumina sequenced it with Jack at University of Chicago. We also then we took it and we sort of vortexed it, purified the viral-like particles with two different methods, all right, cesium chloride and membrane. Membrane actually looked a little better. And then we extracted it, sequenced it, and sent it on. At least that was the plan. That was the plan. All right, so let's talk about the epifluorescent microscopy. Epifluorescent microscopy, so these are 3 epifluorescent pictures from different settings. So you can see the large, sometimes they had eukaryotic cells, the really large things. They have this, you can see by size. It's a DNA stain, DNA and RNA stain, that goes right through the bacterial cells and the virus capsules and glows under UV light. So it's really easy to see and count. This is the standard way used to tell the ratios of bacteria and viruses, all right? So we filtered them onto 0.02 micron filters, stained them with cyber gold, and looked under the microscope with the training of the Forrest Rohr group who do this all the time. Okay, so what did we find about the viral-like particle abundance? Okay, so first of all, we did find that we tediously counted 62 different samples. You do 10 things per sample. This is why God invented graduate students. Are you listening, John? We found viral-like particles. All right, good news. Yes. Bad news, there weren't very many. That's very bad. The bacteria weren't very many either. There's about 10 to the 4 per microliter after we vortexed, all right? And so if you look at these, this is 12 of the samples that I looked at. The blue bars are the viral-like particles, and the red are the bacteria that we counted. Do you notice anything about that?
Comparable.
It's about the same? Yeah. It's almost, in fact, it's a little less viruses than bacteria. Okay, so when we did the calculations, we found a ratio of 0.7 to 1. This is 0.7, not 10 to 1, 0.7. We can talk about that. There's not very many, that's bad. But it's interesting, right? 13 times less than most situations. All right, I just wanted to show you we did a nice bacterial study that actually finished something, but it is exactly the same results as Flores et al. Found. 2 minutes, thank you. We found exactly the same results. We found clustering of communities by site they were taken, the floor versus the soap dispenser versus the toilet seat, a couple of exchanges. And we found that the female and male, the floor itself was the most diverse in terms of the bacterial abundance. So we can do the bacterial-viral ratios if we can only get the viral data. All right. Oh, and I should point out that was only after only 8 hours. After 8 hours every time, repeatedly, we found the same exact results as they did just sampling without any cleaning or anything. All right. The viral purification. The subset of the richest samples, we used 2 bacteria purification strategies. We stopped with membrane because we had a lot of contamination in cesium chloride. We've seen better than that. And we purified it and verified it through microscopy. So I'm going to go through it really careful. And in the immortal words of Elmer Fudd, be very, very careful. If you have to quote Elmer Fudd, you know it's weird. All right, so first of all, we made sure we saw viruses in the sample beforehand. We purified it. We made sure we saw viruses afterwards. I think this is a very important point. And no bacteria. That's great. Then, we still see viruses. We removed the free DNA with DNase because it doesn't go through the virus capsules. We still saw viruses. We think we're doing good. We saw it was free from bacterial contamination, right? We're trying to get rid of the bacteria. So we did a 16S study, no bacteria amplification. All right, then we extracted. We used a mobile cell kit, which does well with viruses. Great. Oh, no, not much DNA. Very little DNA, almost undetectable. What do you do? Well, you do what everyone else does, you genomify. That's often what you have to do with VLP. You take a random amplification of all the DNA, all right? But unfortunately, we just still didn't have much, and this is where the problems begin. We did it again. All right, here's the problem. You have any bacterial contamination. That's what you're going to do. All right, but we did find a few things that look like viruses. So what did we find out? Well, 5 out of the 6 samples worked. That was nice. We sequenced between 2 and 17 billion base pairs per sample, so we're going pretty deep here. We analyzed with MG-RAS, thank you, Jack. But our method is not enriching for viruses. This is the bacterial sequences we found, 0.2% for viruses. This is not good. The viruses did seem like what we expected, though, which is very nice. We did find, of the viral sequences we found a lot of viral functions, phage tail genes, replication genes. It was all there. So we know they're viruses, all right? They could be prophages, that would worry me. But we found it in all our samples. Here's sort of a list of the gene functions, lots of phage. This is a little more encouraging when we looked at taxonomy. We're finding enterobacterphage. Hey, we have enterobacter in those bacteria there. We have streptococcus. What's this? One sample had 9% rhinoviruses. Hey, that's good. That's especially good because they're RNA viruses. How does that work? How do we get RNA viruses on the DNA sequencing? Please help me. Are they like DNA in the human cells on the floor? What's going on? They're going to sneeze and then, I don't know. We're sequencing DNA. I don't know what happened there. All right, so the conclusions. On all surfaces we found. They are definitely there, yes. Not very many. 13 times lower than most situations. Filtration is useful, yield is terrible. Repeated rounds of amplification are probably an issue. We have to do something about that. Low biomass is a real, there's people doing metatranscriptomics on indoor environments. We definitely have viruses. We have some encouraging signs with rhinoviruses. So the next step is, I think, this is what I'm going to try to do. We're going to try to get a lot more viruses. Maybe sample the whole bathroom floor if I have to, really, and sneeze on it. Purify it and concentrate and get enough DNA so we only have to do maybe one round of genomify. And I also am going to check 16S after genomification because I want to make sure after genomify I didn't just get a bunch of bacteria. So this has taken a long time. I appreciate the patience of the Sloan Foundation. I never thought this was going to be easy, but I think we got some viruses there, and we'll see. Wish I had more positive results. But I do think it might be telling us something about these systems, all right? And I'm just going to put my opinion out there, but I think these systems, a dry surface on the indoors, is not a very habitable place even for bacteria. Do we know the activity and the growth of bacteria on these surfaces? And if they're not growing, are the viruses going to be replicating and go through a lytic cycle? Or are they just lysogenic? Do they last long in the environment? Is this a marker for how active it is? And can I go to wet areas and find more activity? I'd like to go. Just go to Antarctica and places like that. I'd like to go near the urinal. Yeah? That's what I'm excited about. The splash zone. Yeah, there might be more activity. It might turn nitrogen fixed, nitrogen oxides. Oh yeah. So I'm thinking of doing a transect across the bathroom, going to places where it's more habitable, a little more carbon, a little more water, a little more excitement. There you go. I gotta switch. Thank you. Any questions? Yes.
So we're actually working on also trying to isolate viral particles in air samples in the Pure lab. We're in the early stages of the process as well, and we acknowledge pretty much all the problems that you're seeing. But just a couple of comments. First of all, with the NG-RAS analysis, you may actually have more viruses than you think you do, just because they're so novel, and the hits that are going to be in the database perhaps are 2 bacteria. So that's one thing that might be worth considering. And then also, lots of viral particles, right? It's really important to look through fluorescence to make sure you're seeing viruses along the way. But there's a trade-off there, of course, so it might be worth, doing samples in tandem where you maybe look under the microscope at one sample and kind of hope that...
Oh, because you lose them? Because you take the material?
When you take a little bit out.
Yeah, that's true. Yeah, we didn't take that much out, but yeah,
right.
I gleefully appreciate that.
Yeah.
Yes. Any will matter.
There are also other amplification protocols
Okay.
that may be worth considering. So I'd like to talk to you.
Oh, we should talk for sure. What was your first one? I remember having a comment about that.
The first one.
Oh yeah, MG-RAS. We just had this a week ago. Sorry.
Yes.
And we realized really quickly. Actually, it's 2 or 3 weeks ago. It took MG-RAS well. We realized it wasn't a great viral database to compare to. We just got the data, but that is an excellent comment. We need better viral curated databases.
So what do you think the 10% bleach is doing? Because you're sampling after you hit something lower number with the 10% bleach. Have you tried just an unclean floor? Not for the bacteria, but for the viruses to see if you get a better recovery?
Yeah, we have done that. It's not a lot better. I mean, we didn't do recovery, but we just looked under the microscope. The ratio is pretty much the same. It's not a whole lot better. I mean, I think I'll just go in a dirty floor. I'm not going to do the whole bleach. That was a special study.
Right.
And that's where we got the samples because we wanted to compare to our data that we'd already generated for 16S. So I agree with you. We should do it. I'm not sure we'll see a lot more, but I want more area, and maybe we'll get a better result. Let's let it get dirty.
And I don't know.
Tell the janitor not to come in.
What's a 10% bleach do to a viral?
Well, we looked under. So I didn't show you the slide, but we tested how long it took for epifluorescence to detect nothing, not a single speck. So we started 2 minutes, 10 minutes, and after 20 minutes, does anyone leave bleach on a surface for 20 minutes? Nobody does. After 20 minutes, we saw no evidence on any surface that we had it. So we really did a test of that, and it was very, very sterile. So we're going to start, if we do a time series, that's how we start. But I think it develops like that. Yeah. One more? One more? Is a question, or I got to get up?
No, I'm just.
He's got to go. Yeah. I thought he-